Hello?
Hey.
Hi there. Okay, everyone seems to be coming on. Wonderful. It looks like we don't have Maya on yet, but she should be coming on soon. And I think she'll be helping facilitate today's class. How's everyone doing? Hey there, welcome.
Hi Taylor, how are you doing?
Hi, I'm doing wonderful. Just got back from the ocean, so I'm
Nice.
very happy. I'm happy that there's sunshine and it's all good all around. Hey Maya.
Hi.
Hi.
How's it going?
So good. How are you?
Good.
I'm a little overwhelmed, but good. Yeah. Got a very full plate this week.
Mm-hmm.
Whew. It looks like you're in a beautiful place. Where are you?
I'm on my back porch. It's the sunshine.
Awesome.
Yeah, it's very sunny here. So I'm absorbing it, using it to its maximum capacity.
Awesome.
Looks like a beautiful day there.
Very beautiful. It's quite hot. And you know, as much as I'm celebrating the sun, there's also a part of me that's grieving it a little bit because we've gotten no rain. It's like the first February with no rain in this area in a very, very long time. So it's very hot and dry. And so there's part of me that's celebrating, and there's also part of me that's sad because there's no fungi out right now. And it's February in Northern California, so there's a little bit of
Okay.
yeah.
There's this word for it. Oh, somebody just told me. No, there's like a word for when something about climate change like is really nice and enjoyable for you, but then you also feel all of this anxiety at the same time about it.
Yep.
There's like this this conflicted emotion that's like really specific to that, you know?
Yeah, absolutely. I would love to learn the word for that because that's all I've been feeling the past like entire month. I'm like, oh yay, celebrating this beautiful weather, but also the ecological lens, like the ecological awareness of that I hold is just like, this actually isn't.
Okay, I think all your weather is probably over here right now.
Yeah.
We've had a really wet summer. It's been amazing, and we've had the earliest mushroom season since 1996.
Wow.
There are fungi everywhere. We've had the earliest saffron milkcap season ever.
Wow.
They're just everywhere. So people are out foraging and getting huge basketfuls, and it's amazing. We're all just like, wow, what is going on?
Yeah.
So
things are definitely, just changing the patterns.
Yeah. Whereas last year our mushroom season didn't start till May. My supervisor was kind of almost jumping in his chair going, there's no mushrooms. What are we doing?
Yeah.
So
I'm grateful that there's wetness where you are. Yeah, and supposedly Washington has had one of the wettest seasons that it's ever had. So
okay.
Yeah, things are just moving and changing all the time.
Yeah. Pacific Northwest, Washington.
Yes.
Washington State.
Yeah, Washington State. Yeah, sorry.
Yeah. Okay.
Sorry to clarify. There's probably a lot of Washingtons.
Hi, Danielle.
Hi.
Hello.
Nice to see you.
Nice to see you too.
So, Kristen, I see you. Are you here still?
Hi, I'm here.
Hey, I just got your email about the poster, and I think that's a really valuable critique. And I wonder if we could just, I'm wondering if you want to just share that with folks right now, some of your thoughts on that while we have a couple minutes before we get started.
I guess just that it looks like there weren't very many replicates if it was just 3 buckets for each of the variables. And
so which poster are you referring to?
Oh, the one on mycofiltration, it had, I think it was Paul Stamets and a few other people looking at the mycofiltration of E. coli.
For stormwater bacteria removal? That one?
Yes.
Okay.
And let's see. Sorry, I have a pair.
The folder, it's called Mycofiltration Salish Sea, is the name of it. Salish Sea Ecosystem.
Yeah. And then also that it says M per D, I think is minutes per day. And so for their experiment, they were like flowing some contaminated water through these microfilters for only like 2 minutes a day. And that's what, and then I guess they would dry out and they would test to see if the E. coli was being captured. So measure the before and after flow rate or flow concentration. And I was thinking in an actual field study, it would be flowing maybe like constantly or, you know, submerged for a longer time than just 2 or 2.5 minutes. And so that was kind of my biggest critique with it. And then also it sounded like they found this pink bacteria, which causes necrotizing fasciitis. And that's really concerning. That was in their wood chips and the straw. And it seemed like maybe it was in a lower concentration in the myceliated ones, but not all of them. There were some that was actually higher. And it was like fruiting from the wood chips. Or like bacteria was growing out of the wood chips. So don't want that to spread.
Yeah.
No, you want to be careful there.
Kristen, Kristen, sorry. Hi. I've seen that happen too. Sometimes mycofiltration setups can actually breed pathogenic bacteria, something to watch out for. But other times, yeah, I've been part of some studies as well for stormwater filtration that were trying things out in more realistic scenarios with much longer and even like very varying, you know, like amounts of time being like pretty totally submerged.
So I have to go for a couple minutes. My realtor's here.
Oh, cool.
So we'll be right back. Hello. Here, 1 second.
Oh, Maya, you're muted, I think.
Oh, I muted myself.
Yeah, I saw your voice moving. But.
Thank you. Hi, folks. Welcome. Wonderful to see you all. If anybody wants to do like the full screen and we can all just say hi to each other real quick.
Hi.
So today we've got, if you press that little button on the top right, you can see the gallery. It's just so great to see you all. Today we have the great honor and privilege of having Danielle Stevenson with us to talk about mycoremediation and toxicology. And she's going to go for about an hour and a half, and then we're going to have you know, half an hour to 20 minutes at the end to talk about how this course is going in general for you, any ways that we can support you in your learning. If you have questions from past things that still you haven't been able to ask, we'll have a chance to ask those. And just make sure that we're all still on the same page and everybody's kind of caught up to speed. So if that sounds good to folks, you can give me a thumbs up. Yay, thanks. And I'll hand it off to Danielle now.
Okay, hi everybody. It's an honor to be with you today. It's a different interface for me, you know, workshop kind of through a screen. So I'm gonna I'm gonna share my PowerPoint with you. And I hope that you have questions as we go through, clarifying questions like, what does that mean? What does that word mean? And other types of questions. So I'm gonna take a few pauses throughout the hour and a half to give some space for questions, but also feel free. I won't be able to monitor the chat box, but Maya's offered to keep an eye on that. So if you do wanna pop a question in the chat box and then, you know, alert me. So, because I don't think I'll be looking at my slides as well, so I might not be able to see you. So just call out Danielle if you have a question, or pop it in the chat box, and I'll also leave time for questions. Okay, I'm gonna share my, yay, okay, cool. Okay, can you see my presentation now? Yes, okay. Is it in the full screen or just not? Okay, so I'll put it in slideshow view. Okay, is that good? Yes, okay, perfect. I wanted to start by briefly introducing myself. I understand there's a real great mix of different backgrounds and interests in this group. So I thought it might be encouraging in a sense to share a bit of my story of how I got into mycoremediation. So I actually was doing food system work and realized that soils are often contaminated, and that can you know, be a big issue in terms of people having access to healthy food and being able to grow healthy food. So that's when I heard about how cool mushrooms are at helping build healthy soil. And I started trying to learn as much as I could, and quickly learned that to work with fungi for remediation, you need to understand how they live and thrive and be able to grow large amounts of mycelium. So I started doing that, and that's how I ended up having a business called DIY Fungi, which like offers mushroom spawn. And also I've worked on a number of different types of mycoremediation projects on different sites, as well as more holistic like bio mycoremediation projects, as well as some more waste management or like applied mycology for dealing with toxic waste. And now I'm in grad school in an environmental toxicology program at UC Riverside in California. So I share that because there definitely was a time when I didn't understand anything. Like learning about mycology and then learning about remediation and toxicology and how contaminants work and what to do about it was all new to me, and a whole new sort of language to learn. And so today but I learned a lot through just doing it in a kind of citizen science, amateur mycology way. And now I'm way sciencey all day in grad school. But today I'm going to try to offer something that hopefully you feel you can take away and try out, or yeah, so I'm gonna try to use a mix of ways of talking about this topic and offer a bunch of, you know, case studies and ideas. And feel free to just, yeah, pop in, call my name, and ask me if you need clarification about something. It is a big topic for an hour and a half, so we'll see what we get through. Thank you. So I want to start by defining mycoremediation. It's about working with fungi to break down and or extract contaminants from the environment. And they're already doing this. Fungi are already doing this and have a whole range of natural abilities. These natural abilities are what we're trying to support when we're working with fungi to deal with toxic land or water or materials. So some fungi have a natural ability to degrade organic contaminants, including petroleum products, agrochemicals, and turning those into less harmful substances, or starting a process of breakdown that other microbes and organisms can finish up, ultimately creating soil usually. Some fungi have natural abilities to immobilize toxic metals in the soil or extract them through hyper-accumulating them in their mushroom fruit bodies, or helping their plant partner hyper-accumulate them as part of a phytoremediation strategy. Some fungi also can immobilize biological contaminants like E. coli. And in general, fungi play really important roles in building healthy soil and supporting many other life cycles that are important to the regeneration of ecosystem health. So there's three main sort of streams or potentials with mycoremediation as I see it. We can work to remediate and restore contaminated sites. We can reduce the toxicity and accelerate the decomposition of polluting wastes. And we can clean up after a disaster or a spill or a pollutant release. So I drafted this really simplified overview of some project steps, how to get started working on a mycoremediation project. And the presentation is sort of organized going through these three main steps one at a time. So just by way of overview, the first thing you want to do is learn about your site and contaminant. So assess the site, test the soil, the water, the material, research all of those things, build your team, and set goals. You want to then come up with a plan, choose your approach wisely. We'll talk about best practices and things to consider in planning your mycoremediation project. You'll need to identify and gather the materials and resources you need, and then make sure you come up with a plan for how to responsibly store and or dispose of any toxic byproducts of your remediation. Finally, you'll implement your plan. When you install, you'll protect yourself, maintain and monitor the site, and then retest and figure out your next steps. So first of all, it's important to understand that different kinds of contaminants need different remediation strategies. So some questions to ask yourself are, what contaminants are present and in what concentrations? Is it a mix of like organic chemical contaminants? Is it, and inorganic ones like metals? Is it multiple of each kind of contaminant? Do they need to be removed from the site for treatment, or can they be treated on site? And should they be contained during treatment so that they don't leach off or move from your site and cause harm somewhere else? Again, how will you take responsibility for any potential toxic byproducts of your remediation strategy? And just consider the long-term impacts of your design. And really important to consider what the site conditions are and what the environmental conditions necessary for the fungi you're planning to work with to survive and thrive. So some general categories of contaminants to understand, and we'll go into some examples of these. So first off, the organic. They're called organic. That means carbon-based. They're carbon-based. They're manufactured or derived from something that was once alive. That's what that means. And there's a lot of examples of these. Some of them you might have heard before. So we're going to talk about PAHs. That stands for polycyclic aromatic hydrocarbon. There's a bunch of solvents. You may have heard of POPs, which stands for persistent organic pollutant. So these are the kind, they're persistent, they stick around and they bioaccumulate. So an example in here is DDT, the commonly used insecticide, and PCBs, which are the kind of contaminants then end up in whales, like for instance, orcas having huge amounts of these toxins. They build up and they don't break down. Other examples would be pesticides and herbicides, like all the ones you hear about, Monsanto using atrazine, glyphosate. And then there's some newer so-called emerging contaminants, like the PFAS chemicals. They're used in firefighting sprays, but a lot of other products. And yeah, we can talk about any of these more and remediation options if you're interested later. But the thing to know about any organic contaminant is that they can actually transform. They can be broken down or degraded into less toxic or non-toxic forms. And usually we're working with decomposer microbes to degrade organic contaminants. So I had sent a Agency for Toxic Substances and Disease Registry article. They're a great resource. They have a lot of just 2-page talks, like frequently asked question sheets about every possible kind of contaminant. And the one I shared with you, I think, was about total petroleum hydrocarbons. But you can, if you want to know something about a contaminant and how it moves in the environment, how it affects human or ecological health, you can look at it. I recommend that database. So any petroleum products like gasoline, diesel, originally come from crude oil. And they're complex mixtures of hundreds of compounds, which petroleum hydrocarbons are one of the main components. So sometimes when I teach this, I actually pick one, I think I've picked diesel in the past, and actually like listed out all of the compounds within diesel oil, and it takes like 8 slides to keep scrolling through all the different compounds. So a lot of other products are actually manufactured from petroleum hydrocarbons. So a lot of pesticides and herbicides and other products as well.
What was the resource that you were just talking about where this information is found? Can you
the acronym is it's the Agency for ATDSR, toxic. It's like Toxic Substances and Disease Registry. It's the fact sheet I had sent out. There was a 2-page one. I think you included in the homework or the Google Drive. I have a link to it again in a few slides if you want.
I'll share it.
But it's just great because they communicate really clearly and simply. Like it's just a 1 or 2 pager about each, and then links to all these other readings you can do if you want. But I see the chat box is going off.
Yeah, that was a question that hasn't been addressed yet.
There is?
That was the only one.
Oh, that was one. Okay, thank you. So the reason I'm talking a bit about some of these key contaminants is because actually the physical and chemical properties of each contaminant are really important to the harm they might cause to human health, and also the remediation strategy that will be effective. And so this is a picture of a distillation chamber. It's just like how we make different types of gas and diesel and oils from the crude oil, the really heavy tar crude stuff. And yeah, it's just important to understand, like I'll pick a couple examples from here. So this is relevant. So say that I've worked on an oil spill response before, and the spill was in a fast moving cold river, and it was from a pipeline rupture. And so what you'd expect with a spill like that, an oil spill, is that the lighter, so like the petrol, like the gas, and some of the like benzene, toluene, jet fuel is also quite light. So those lighter components would quickly volatilize. So that would be something to know, because they would get into the air and can hurt people's health. And the heavier stuff is more likely to actually just sink. It's heavy, even though oil generally floats on water. Really heavy crude oil is likely to sink, and that's a problem. And it's relevant when you're doing a remediation strategy, because that like heavier oil that sticks around longer can end up causing more long-term harm in ecosystems if it's not resolved. So you're going to need to consider this as you move forward with remediation strategies. Polycyclic aromatic hydrocarbons, PAHs for short. I had sent an article about these as well, and their remediation with microbes and fungi. They're a really big deal. They're all over because they're what results when fossil fuels are burnt. And so a lot of the mycoremediation studies that have been done so far have looked at addressing polycyclic aromatic hydrocarbons and also like diesel. So PAHs are cancer causing. They are often found, any coal plant is going to have a lot of PAHs around, alongside of any busy road. Anything involving fossil fuels being burnt, there's going to be PAHs present. I don't know, Mia may have mentioned PAHs are also a common contaminant resulting from forest fires. So it's a good one to know about. And here is the link to the Agency for Toxic Substances and Disease Registries fact sheets, if you want to learn more about any of these contaminants. Metals. So metals are elemental. They're not organic. They're naturally occurring, but they don't come from something that was once alive. So examples include lead, arsenic, cadmium, chromium, mercury, and so on. They're what you see on the periodic table, if you remember chemistry class in high school, whatever. So the important thing to know about metals is that they cannot be broken down for our purposes. They can't be broken down like organic contaminants, but they can be immobilized or extracted so that they're causing less harm. I also want to highlight that there's some really common pollutants that seem small, but cumulatively have a large impact. So, oil leaks, animal, like pet waste, cigarette butts, lubricant oils, household cleaners, septic systems. There's a lot of, and I mention this because this is a way that I started to get into mycoremediation, is working on smaller scale mycodigestion of common waste that I was connected to in some way to try to prevent them from building up in the environment. And just a note on that too, because you had a section on mycofiltration, I'm not focusing on mycoremediation of biological contaminants in this presentation, although definitely that's something that is possible that people are working on as well. So how to test for contaminants? I will just go quickly through this, but I can include this in the resources after. This is really important. It's important starting out to test what's there. As many samples as you can, and for as many types of contaminants as you can. For instance, there are actually metals, toxic metals in a lot of petroleum products. So that's important to test for and consider in your remediation strategy. Most commonly, people will send samples to a professional lab for analysis. This is super legit. If you're trying to work on a experiment or project that you can, you know, confidently share with others after and say, hey, this really worked. You know, the contaminants were in these concentrations at first. We extensively sampled after our treatment with fungi, you know, they're below these, we reduced them significantly, check it out. You know, we sampled all the way through, we saw the breakdown, now there's less contaminants. So it's really legit, it's really expensive. I've had luck over the years partnering with university labs to access testing. Often there's students who might be working on something similar and would love the chance to help someone out who's doing a real world application that's, you know, trying to solve a problem. So you can try partnering with university labs. There's other ways I've listed that you can save on testing costs, like you can test just for total petroleum hydrocarbons. That just tells you, and actually a lot of the studies we'll talk about have done just that. So that just it doesn't tell you there's this concentration of this type of PAH and this type, and there's this many, there's this much of, you know, diesel and so on. It just tells you, hey, there's this much hydrocarbons in general. And so it's very crude, so to speak. You can see that you've reduced the total petroleum hydrocarbons if you test, if you just have the budget through fundraising or whatever to do total petroleum hydrocarbons at the beginning and end of a study, that's definitely better than nothing. There's also some really cool, just more citizen science ways to test and see if your remediation strategy is working. And so there's some, they're called, there's like field colorimetry and reagent-based approaches. And I have some links to, like, Public Lab is a great organization that shares like ways to work with these kind of methods, like super cheap, super DIY. We still need a lot more people to work on this and share their findings so that we can better be able to match like what we're seeing in our samples to understand concentrations of certain contaminants. And then another option is working with bioassays. And this is because some living organisms like worms, nematodes, beans, lettuce have known thresholds to certain contaminant concentrations. So above which they may die or not be able to reproduce. So I have some articles and protocols I could share for this as well, working with earthworms as a bioassay, where you can, again, you're not going to be able to say, you know, we had this much of these contaminants before and now we don't have them anymore, but it does give you a pretty good sense of whether you are reducing toxicity through your mycoremediation treatment. So tips for taking samples, I've talked about a bit, but the more you can do, the better. Spread out over your site or your reactor or whatever you're working with at different depths. If you can test for the full suite of toxic metals as well as the other contaminants that might be present, that'll really help you design a really awesome remediation plan. So I'm going to go into next how to protect yourself when working on remediation, but be sure to do this. If you're working on a toxic site or with a toxic material, don't hurt yourself for science or for any reason. Yeah, so at the very least, like, that's why it's always good to start by researching the contaminant, understanding its health impacts and how you could be exposed to it, and wearing, you know, at the least gloves when you're doing sampling and doing work. I'll just send some info on like how to take and store samples. If you're testing for organics, you need to understand that because they can transform, they can break down, you have to store them properly and get them to the lab very quickly so that you get accurate results. It's not true if you're just testing for metals. So understanding where the contaminants have come from and if they will keep being deposited is really important too, because contaminants can actually end up on your site through the air, through water, through nearby polluters, or either historical or present land use, and of course from like disasters or a spill or release event or a flood. So if you, I mention this because if you are working on a site, say that you know the site has diesel contamination because it's been a transit yard for a long time, but the site happens to also be like next to a coal plant, you know, you can remediate the diesel that's built up in the soil, but you're going to have to consider that there's going to be ongoing deposition from the air, from the coal plant, of like PAHs and possibly metals. So that's why I mentioned that part. Understanding where the contaminants may go is really important to developing a really responsible remediation plan. So this is also known as fate and transport, and it has to do with the properties of the contaminant and the site characteristics. And so what I mean is you want to be conscious of, for instance, how water moves on the site so that you can adequately make sure that whatever contaminants are on the site you're working on aren't going to leach, like when it rains or whatever, floods even, that they don't leach into the groundwater or run off onto a nearby site. So you'll want to assess your site thoroughly. Look at the soil characteristics, the slope, how water moves on the site, like I said, who uses or passes through the site, all different types of animals, soil organisms, what plants and microbes and mushrooms are already there on the site. How does the sun, wind, and other environmental factors affect the site? And what is its history, like culturally, land use history, and who's around currently? So to protect yourself when you're working on remediation, it's important to understand the contaminants you're working with, like I said, and how you may be exposed, and then protect yourself with the right protective gear. So you can, in general, depending on the contaminant, be exposed to contaminants through your skin, so through touching the soil, the oil, whatever it is you're working with, through inhalation. And so this is really important because if you're working on a site and it's windy or dusty, for instance, you can actually, or you're working on an oil spill or something, and there's some of the lighter fuels that are volatilizing, they're getting into the air and you're breathing them in. Inhalation is kind of an unseen way that we can get harmed through working on remediation. And then of course, like ingestion. So often this is unintentional. You have some of the contaminant on your hand and you touch your mouth and you eat your sandwich with your hands later. So you can get more training. There are some, I put a picture of the hazmat school. So they have a whole range of, it stands for like hazardous materials. So if you are really seriously wanting to go into doing remediation really seriously, then it would be good to get some hazmat certification. Okay, so before we move on to the next section, coming up with a plan, does anyone have any questions about that last section?
I saw a question that just came up about when is it not worth remediating? Which is, I think, is a fabulous question. You know, like, when do you go in, when do you intervene, and when do you stand back? And there's definitely a time and a place for both.
I'd love to hear other people's thoughts on this actually as well. I think it really depends on the situation. I think in the next section I'll talk about, I generally think that we already have a lot of sites, a lot of soils and waters that have been contaminated through past or ongoing industry and extraction. And we also have times when a disaster occurs. So there's like a hurricane and it causes flooding, which means that every, the wastewater treatment plant and like the oil refinery and like every contaminant ever gets like all put into water and then spread all over a land. Or there's a mine tailing rupture and then toxic metals move everywhere. So in the area. So there's kind of different, I think that it really depends on the context. I think generally, if there's a disaster or a spill, it's really important to get that toxic stuff out of the water and out of like water, or like stop it from continuing to spread, and then contain it so that it can be remediated. With legacy contaminated sites, perhaps probably that there's already a lot of life present on the site, and I'll talk about that more, but starting from, no matter the level of contamination, like supporting what's already present on the site, doing the work of healing is a good way to go. And then having said all that, like I also think that sometimes when I first started with this stuff, I like really resented the conventional remediation approach of digging up contamination and moving it somewhere out of sight in some way, and not dealing with it. Or commonly also, like, sites will just be capped, so there'll be like a clay layer or just some sort of like fabric or material that basically just holds all the toxic stuff down, supposedly, and then they'll put fill on top and even, like make a school or a park or like something like this, and that can lead to a lot of problems. But also sometimes if it's preventing harm and like containing it, then I think there is a place for harm reduction in that way. That was long-winded. I love the question. What do you all think?
I think I agree that if there are native, like autochthonous bacteria and fungi that could clean up that contamination, then there would be maybe bioamendments, but not necessarily mycoremediation that's needed. Another thought I had was, what if the contaminant is a kind that if you break it down partially but not all the way, it's actually more toxic? So up here in Alaska, especially in Fairbanks, we're having a lot of PFAS and PFOA contamination, and those are very difficult to degrade. I think fungi are like the only thing that can break them down, but it has to be really controlled so that it's done completely. Otherwise, you're just making more of them. Yeah. And that's, like, I think kind of out of the scope of most DIYers. Does anyone else have experience breaking down those contaminants?
I hope we can. That is something that we're looking into right now, is dealing with PFAS specifically. There's definitely a gap in the research that's needing to be filled.
I will talk at the end. I did work on a project involving a lubricant that actually is a Teflon based, and so I will talk about that later. working with fungi for that. But I appreciate your point. It's really important this is why actually like research is a really great way to start. Learn about the contaminant and what its breakdown products are. Often fungi are good at initiating a process of breakdown, but sometimes that can, as you say, actually create more toxic products. So it's really important to do research as a starting point so that you design a wise strategy that is responsible and doesn't actually create more toxic stuff by accident.
I think this is such a juicy question. I have just one thought on it, and then something I wanted to reiterate that you said, Danielle. I think that so much of it is about knowing what your capacity and what your ability to really follow through on the project is, and how well you're able to deeply listen to the land and connect with the land and the people who have lived there, the people who maybe know more about it than you do, and also just your proximity. And if you're going to be able to like check on your project consistently, or if you're hyperaccumulating heavy metals with mushrooms or with plants, are you going to be able to come later and remove those and put them somewhere safe, or are they just going to grow and then the heavy metals will go back down into the soil? So that's, I think, super key, is to just really think long term with all of these kinds of projects. And Danielle, I know you said this, but I just really want to put an extra emphasis on how crucial it is to both get samples before and after and gather data when you're doing these projects, even on a DIY level. Like, that data is precious to us all in the applied mycology field. And I can't tell you how many projects have happened where it was great and exciting and we thought it was a good idea, but we never got any actual data out of it. And if you ever do collect that data, send it to us, share it with us. We also, sometimes we have access to people at universities who can test things or look at things or might have another perspective on things. So yeah, I'm seeing a couple more really juicy questions come up. Chanel, I'm wondering if you want to just ask your question. Are you on mute? Chanel, are you here? All right, maybe we'll do another. Oh, your voice doesn't work. I'm so sorry. Okay.
Does it work? Wait, can you hear me?
Oh, no,
yeah.
Oh, it does work. Okay. Hello. My question was about, because I know you talked about how it's important to also check if there's other fungi present before you even start it. So what happens? For example, sometimes you might not know, or something that you may miss. What happens when these things like join together? Like, I don't know. I don't even know if that's a proper question. I'm still learning, but like just the emergence of like two different fungi and like that, like, yeah, how will that kind of like play out in the world or in that project?
Yeah, that's a really great question. Thank you. I think we will talk about that a bit soon. So like, kind of like what happens when you introduce a new like fungus into a site or whatever, and how that can interact with the native, like what the fungi and other life that's already there. And then also like what are called like strains. Like there can be these special strains of a fungus that you can work with the fungus to develop that are more able to like do a great job at eating whatever the contaminant is or thriving in hard conditions. And that occurs naturally, but we can work with that as well. Does that kind of get into what you're asking about, Chanel?
Yes. Also,
okay, awesome. All right, if that's cool, that's great. We're gonna actually go into that in the next section. Thank you so much.
Real quick, is there a difference between having lab created like mushrooms, like kind of like things I figure out in the lab versus like the natural ones? Is that also a thing and part of that?
Oh yeah, you're totally onto something. And it's a big part of what's going on in mycoremediation right now, is that there are these a lot of the like studies that have been done were done in a lab in like perfect conditions where the fungus never had a hard, you know, moment in its life. It had everything it needed, you know, did an amazing job at degrading a contaminant in a short time. But then there's very few studies where fungi were actually applied in like real conditions. And usually when they are, it doesn't work out very well. And it's because there is a difference for sure, which gets into, yeah, the next section is all like best practices, like what we've all learned and how we can do things differently. Like I propose actually starting with looking at who's already there on the site or on the contaminant, and then amplifying what they're doing, supporting them in what they're doing, rather than introducing a fungus from that maybe isn't from that place, and therefore might be risky ecologically, but also like just might not be very adapted, might not be able to survive well in that environment. So that's perfect for the next section too. Was there another question, Maya, before we?
There's some questions. Are you going to talk about agricultural contaminants later on in this talk? Okay.
Yeah,
great.
that's actually my research. I forgot to mention that. I have a couple, like one slide on that, but that's actually what I research, is how to work with arbuscular mycorrhizal fungi to support phytoremediation, but also to prevent toxic metals from getting into our food. So I'll talk about that later.
And then we have one question here about whether there's a test for heavy metals for mushrooms specifically, not just in soil, which I have no idea. I've never heard of.
Yeah, actually, I just did that. Okay, so if you have, so my lab that I'm in at UC Riverside is looks at metals in soil and water, and so we have all the ability. We do a lot of testing for metals, and last week these grade 8 students contacted me and were like, hey, we did this project where we spiked these oyster mushroom kits with these different toxic metals, and then they grew them and harvested the mushrooms, and I analyzed the mushrooms and saw like to what extent those toxic metals were getting moved from the mycelium into the mushroom fruit bodies. And so it was super cool. So if you have metal related projects, yeah, you can definitely, contact me. Happy to help with that. You basically just need to really, really thoroughly, I had to freeze dry because mushrooms have so much water in them and that throws off the equipment basically. So I had to freeze dry them and then powder them. And then I used an XRF, it's called, to analyze, to see each sample the total concentrations of metals that were in the mushrooms.
Amazing.
Yeah, I was so excited that these grade 8 students were, I was like, yeah, you're helping answer an important question with mycoremediation.
What did you find?
Yeah, so copper, so they just used copper, silver, and iron because they did this research and found out that those are metals of concern after there's been a forest fire, and that they're in California, so there's been lots of fires. They heard people wanted to use mycoremediation and that this wasn't really understood, and so that's why they chose those metals, I guess. But so we found that the copper and the silver were very much like accumulated in the fruit, in the oyster mushroom bodies. I'll share about it. Like I asked if I could share, they're doing it for a science fair. And so once they have everything together, they said they'd be happy if I shared it with people. So, but yeah, they were accumulated, not the iron. But anyways, that's a whole a whole side thing. Are we ready to go into the next section? This is the bulk of the presentation.
I think we should go for it.
Okay, cool. Yeah, it's so funny, I guess, because you're all muted. So I was like, oh, okay. Okay, so starting off right away. So this whole next section is about coming up with your plan. So you figured out what the contaminant is, you've learned about your site, you've done a bunch of research, now you're coming up with a plan. And so I'm gonna offer a bunch of different things to consider and best practices based on what's out there in the literature, based on what I've seen from my own experience and through others in the network. So first of all, like we've been talking about, amplifying native fungal species works better, just actually works better than non-native species, because they're already there. They showed up, they volunteered to digest that contaminant, to live in those conditions with all its, you know, whatever the temperature and climate are, you know, whatever the amount of moisture available, everything like that. So a really great idea, I think, and I've seen in the literature and I propose, is start out by collecting the fungi, and not just the fungi, collecting the plants, the bacteria, the fungi, the life that is already growing on that site. And it doesn't have to be a site. If you're working with a certain toxic material, like cigarette butts or something, or you know, whatever it may be, literally just like gather that, gather some like dirty cigarette butts from the environment, and then learn what bacteria, fungi are living on that because they want to. They chose to show up there because they think they could try to eat it in some way. So I really propose starting here. You typically, this protocol, this diagram is from the Thomas et al. Field demonstration of mycoremediation study that perhaps the speaker who's about mycofiltration brought up. It's a neat study, but this is from there, and it's just showing the process where you collect the fungi, isolate them to get a pure culture, is their proposal. And then you just continue, as you learned in the mushroom cultivation section with Willoughby and others, you keep expanding that mycelium until you have enough to work with on the site. So this is because you're trying to, you got this fungus from the site, or these many fungi or these many bacteria, and you're trying to accelerate the work they're already doing. And so to do that, just you're growing their mass and then reapplying onto the site. And this is another, the next thing I want to suggest is thinking beyond just the charismatic decomposer fungi. We will talk about those a lot. Most of the studies that have been done have been with, you know, the Pleurotus austriatus, the pearl oyster, and some of the other decomposer mushrooms that are easy to see and easy to work with. But just a reminder, I think you covered this in your fungal biology section at the beginning. There's so much diversity in the fungal kingdom, and there's a lot we don't know, but a lot of promise and potential in a lot of other categories of fungi. So the decomposers are a go-to for organic contaminants because they break down dead plants. That's what they do to get their food through their enzymes, and we'll go into that more. And so, like a lot of petroleum hydrocarbons are ancient dead stuff that a lot of decomposers can just break down using their same enzymes that they would if it was wood or leaf litter or whatever. But there's also a lot of cool work coming out about endophytic fungi. so the fungi that live inside of plants, and mycorrhizal fungi as important remediators as well to consider. So we'll go through some examples. I just thought this was a beautiful diagram because it shows you, I think it's just one of the clearest diagrams I've seen of these different types of fungi and where they live. So you might have heard about the Pestiloteuopsis microspore. So this is an endophyte that was isolated from a plant in the Amazon that can survive off of polyurethane plastic as its sole carbon source, even without oxygen. So that's pretty cool. That's what I mean, good to kind of look beyond just the decomposers. A lot of other types of fungi can also decompose toxic stuff. Actually, also a lot of molds and yeasts that are very, very common can also degrade plastic, other types of plastic through hot composting processes. And so some, like Aspergillus and Penicillium, are pretty, there's a bunch of studies about them as remediators as well. This is another article I sent out. I thought it was really cool because it really exemplifies this approach of first, like, starting by looking at who's already growing in really hard conditions on the contaminant you're interested in remediating. So the author, the scientist here actually saw a dandelion growing in pure coarse tailings in the Athabasca tar sands in Canada. And so obviously caught their eye because nothing else was visibly able to grow in those pure coarse tailings. And so when they took it back to the lab and checked it out, they found that the dandelion was able to survive in those really, really harsh conditions that it wouldn't normally be able to because of this endophytic fungus that was helping it grow, and also acting as a decomposer and actually breaking down some of the hydrocarbons. So they called that endophyte TSTH, and then applied it, like basically inoculated, grew it out, and then inoculated plants. Both plants that were used, they inoculated like a remediation mix that it had not been able to grow in the coarse tailings unless it was inoculated with this helper endophyte fungus. And also found that when applied to other kinds of plants, that this endophyte could help the plants grow. So there's also been fungi isolated from, like, you know, electronics waste, which is pretty wild. So some of these fungi are now used to actually extract useful metals like gold, silver from electronics waste. But some of these fungi that have been found growing on different types of waste are also used to filter wastewater and in a whole bunch of other remediation contexts. So all that to say, if you're yeah, whatever it is you're working with, whether it's a contaminated site, a toxic waste product, probably the best fungi and other microbes to work with are already present on the material or the site that you're interested in remediating. And so that leads into the next best practice: choose the right fungus for the conditions. And so this gets into what I think it was Chanel who had asked a great question. Kind of gets into some of that, where it's like, yeah, you need to consider that a lot of the studies that have been done were done in lab conditions. And then when what we learned in the lab was applied in the field, it didn't work out usually very well. And that's because that fungus wasn't able to tolerate the conditions of the site. You remember from your cultivation and fungal biology, a lot, like most fungi need oxygen to live. Not all do, and not all do all the time. Most need moisture at certain levels. Most wouldn't like to live in like full sun. So there's a lot of conditions that are present on real contaminated sites that are not suitable for most living fungi. And so, you know, there are studies about turkey tail pictured here. It's one of my favorite mushrooms. Apparently in the lab, super good at degrading those PCBs. But when you apply turkey tail but PCBs in the real world are often found beneath electrical capacitors and, you know, in soils all over. And in those real soil conditions, attempts to sort of inoculate turkey tail didn't really work out. Like they did not degrade PCBs in real soil conditions where PCBs are found. Some types of fungi work better if what you're dealing with is metal contamination. So mycorrhizal fungi that are symbiotic with plants, most all plants, and associate with the roots of plants are good ones to work with if you have metal contamination. They can immobilize, I'm gonna talk about that later. So I'll just mention that now. But often with organic contaminants, we're usually looking at decomposers, endophytes, and molds. So common molds, as I sort of mentioned, like Aspergillus, Penicillium, and Trichoderma. I have a anecdotal story about how awesome Trichoderma is at helping with mycoremediation later on. As I mentioned, introducing non-native organisms can have risks. In the article about PAH degradation by fungi and microbes, it mentions that like Pleurotus austriatus, which is probably the most go-to, like when, you know, community or citizen kind of mycoremediation projects are happening, it's easier to work with pearl oyster, so people usually do. But unfortunately, pearl oyster is known to inhibit native microbes. So if we apply the pearl oyster on real sites, it can actually like take away space and from native microbes that end up making the remediation less effective, because the oyster is good at starting things breaking down, but it can't do the whole job. And it's actually a lot of those common molds and those common yeasts and those other common bacteria living in soils that are needed to finish the breakdown. So another thing to consider, and leading into the last point, that mimicking nature by working with multiple species in succession is the most effective. It just actually works way better and it makes sense. Cool. So for dealing with metals, mycorrhizal fungi are the way to go. It's hard to be brief. I love mycorrhizal fungi. That's what I study. So, but I'll keep it brief. I mentioned there's two main, so there's different types of mycorrhizal fungi, as you may know. The ones I study are the arbuscular mycorrhizae. So they're like the generalist mycorrhizae, and they have helpful applications in phytoremediation strategies. They both can help their plant partner, which you like survive, like be able to even live in highly metal contaminated soils that have poor conditions. But they also can help that plant extract more metals from the soil. So typically in phytoremediation, we're working with what are known as hyperaccumulator plants that are known to just like be really good at taking up a lot of metals into their plant bodies out of the soil. And when inoculated with AMF, they can actually do that even better. But weirdly, very complexly, application of these arbuscular mycorrhizae can also actually reduce toxic metal uptake in plant partners. And this is really important in agriculture, where actually a lot of the common fertilizers and amendments that we use, as well as irrigation, continuously deposit toxic metals like cadmium into our agricultural soils. And cadmium and other toxic metals are highly available to get up into the actual plants that we then eat. So, but it's really interesting that actually these AMF, these fungi can actually decrease not only the need to apply the toxic fertilizers to help grow crops, but also to reduce the toxic metal uptake into the crops. Okay, so of course we will talk about the decomposer fungi. These are the ones typically applied for organic contaminants and for some metals as well, like oyster mushrooms and button mushrooms and others actually also can be used to, typically those work less by immobilizing like metals in their mycelial mass, and they can like hyperaccumulate them in their fruit bodies. They can do it. They can do all of it, I guess. So you know about the decomposers, but the important thing I want to point out here is that you may have learned there are different kinds of decomposers. There's the brown rot fungi, which can degrade cellulose, and the white rot fungi can degrade lignin and cellulose. And this is really important in remediation. Typically, it's the white rot fungi that have the digestive enzymes powerful enough to degrade the organic contaminants that we want to degrade. So this is a list. I usually don't like giving like a list of like, and then use this mushroom for this, because I think it's more like, because of all the reasons I've mentioned. But these are the white rot fungi that are most well studied. And so how do you say, Phanerochaete chrysosporium is the it's like a crust fungus that's a white rot decomposer. It's pictured, I'm like pointing, but you can't, it's pictured here. You've probably seen it, but it's one of the most studied remediator fungi, as well as even shiitake mushroom and oyster mushroom, of course, as you know. My proposal is that we consider only working with these like non-native, if they are non-native to the area and you didn't discover it on the site, that you consider working with these just in closed systems. So like a toxic, I call them mycodigester in a digester or a bioreactor, and just limit their potential to inhibit native microbes and organisms. Some of these are highly effective at breaking down organic contaminants, and so in some cases it makes a lot of sense to work with them, but you can have a lot of success working in contained systems if you're going to work with these fungi, and that's what I talk about at the end. I wasn't sure how much you would all want to talk about like how this actually happens, like how do these decomposer fungi degrade organic pollutants. My exams and stuff right now, my classes, like I have to look at chemical, like just draw out like chemical structures all day with like how they break down. And I know it's really daunting to look at. But the main point is that I thought this cartoon was kind of cute. It's like, I've heard wood is very tough stuff because it's made of lignin. So how do fungi break it down? And then the fungus is like, we release special substances called enzymes from our hyphae, and they do the work for us. So the breakdown occurs through these extracellular enzymes that these fungi put out into their environment, the same ones that they would use to break down wood or cellulose, also can break down certain organic contaminants, and it's because of how similar their chemical structure is, and the fact that these fungi are recognizing that they might be able to get some food out of it. So originally in one of the articles about pH breakdown by fungi, this is actually showing the, I don't know if you can see my, where I'm pointing, but this is showing the actual breakdown of phenanthrene, which is one of the pHs by Pleurotus austriatus. But that's like kind of hard to look at. I thought this like really fun party time picture was pretty good too. So the POP, that just stands for persistent organic pollutant. And you see the fungal hyphae in the soil. And so some of the main enzymes that these fungi secrete are lignin peroxidase, manganese peroxidase, and lacase. And these enzymes basically like start I don't know how chemistry you want to get about it. They basically enzymatically combust these complex structures and snip apart. I'll go back to this other picture. In the top, we're seeing lignin, and we're seeing it go from being a more complex thing to getting just snipped apart. All the rings are getting cleaved. They're getting cleaved by these enzymes that oxidize these rings. Oxidizing, you might have heard about free radicals with regards to our own health, right? Free radicals are these free oxygens that like tear through and like are stealing electrons and wreaking havoc. And so it's through these enzymes that create free radicals that cleave apart these complex structures like lignin or like a polycyclic aromatic hydrocarbon, and then extract the carbon basically that the fungus wants. So they don't finish the process, but they start snipping things apart. And you can then, and we'll talk about what happens next. But an important thing to know is that this only happens when the fungus is like healthy and alive. These enzymes are secreted by living fungi, and the degradation of your organic compound can be affected by the environmental conditions like the temperature, the oxygen, other nutrients available, the type of the fungal strain, which we'll talk about, the nature of the PAH or the contaminant. So it's important to consider all of this. You got to take care of the fungus that you're working with for it to do the work you want it to do. So a couple examples of this being applied. One of them is by Fungia Farm in California. Maya, are you familiar? Is anyone connected to this project or farm in any way?
I've just heard great things about them.
Okay. Cool. Yeah, so you can read their, okay, cool. Yeah, I put the link. You can read their full report. So I guess like 4 years ago, they were invited to work on this project because a whole bunch of diesel fuel was spilled on a site really close to a river. and the land managers and the watershed council were concerned about the diesel getting into the river and causing harm. So they invited the Fungi Farm to design and implement this mycoremediation project to break down the diesel. So basically what they did, and it's my favorite part about this, is that in their report, they are humble about everything that they would do differently next time and what they learned. And so I really recommend checking out the report, especially the part where they're like, hey, we would recommend that if you're gonna do this, consider this or do this. But the main thing that they did, so the diesel contaminated soil was all evacuated. There were 70 yards of it. They basically started out by making these burlap sacks. You might have heard of them as like bunker spawn, that were inoculated with oyster grain spawn and into pasteurized straw. And then they basically like layered, as you can see in the pic. Well, first up top, you're seeing just the myceliated oyster bunker spawn. And they basically like layered this in with the contaminated soil. And they did think about the conditions that would work for the fungus. So they started in fall when it would be cool enough for the mycelium. But they ran into some problems because the mycelium actually drowned when it started raining in winter. The way it was set up was, it became anaerobic. There became like, they made it too high, and then there were huge sections, like the bulk of the soil in the middle had no air, and this is a aerobic organism. So it literally like got too wet and then drowned, and then huge parts of it, there was no metabolic action because it was too wet. The fungus wasn't able to live. So they tried again, and this time they just inoculated burlap directly. So they got these huge rolls of burlap and inoculated them. And this time they inoculated, like, the thing to think about is if you're doing this kind of approach, you need a huge amount of mycelium, and that's like a real challenge. So people will often see if they can get spent grow kits or like grow bags from mushroom farms, like from bigger farms that are probably going to compost or throw out their kits after they've gotten a bunch of fleshes off of them anyways. So this time they inoculated the burlap directly, and then they layered in straw, like untreated straw, as a easier food for the oyster mushroom to eat, and the contaminated soil, as you're seeing below. This time, basically, they also moved it, and originally they had it in full sun, and that was another thing that harmed the mycelium. And this time they did their piles in the shade, and that worked a lot better. They made sure the temperature stayed between 65 and 75 degrees. They kept the piles less high, so they were less than 4 feet tall, so that they wouldn't have these big anaerobic sections where no degradation would happen. And then they ended the process by adding a whole bunch of compost tea to finish the breakdown. And so they did have some success with this. They said they had a 90% reduction in the hydrocarbons over a 2-month period of this study. And they, as I said, they had a lot of really thoughtful tips for others who might want to apply a strategy like this. So that, and I thought that was really humble, because we all benefit from learning from their sort of trial and error. You've probably all, I imagine most of you have seen some of Paul Stamets' work. Like, for me, to be totally honest, like 8 years ago or 9 years ago, I saw the TED Talk, like 6 Ways Mushrooms Can Save the World. And I was like, oh, cool. Like, I didn't know anything about mushrooms at that time. So, but it's interesting to really look at that actual study that Paul Stamets was involved in. He was the spawn provider for the study that was done by Thomas in 1998. And it's really interesting to learn from like actually looking at the details. So they did report like a 93% PAH removal in the 3 to 4 month period. But they had to use a huge amount of mycelium. So they said they used a 25, like, and all the numbers change when you read the actual study and then you read what is reported out. But they used a 25 to 30% inoculation ratio. And they used, their treatments were done in these 10-yard piles of the contaminated soil. And so 30% inoculation, like 30% spawn of a 10-yard pile, that's like 3 yards of spawn. So that, like, I've been a landscaper and a spawn grower, and I can very tangibly imagine 3 yards of spawn. That's a lot of spawn. So something to think about. So they used a lot of spawn, and they did find a reduction, but they reported actually that one of their other treatments was a compost treatment, and they actually found the most success was when they first did an oyster mushroom treatment and then a composting treatment. So this goes back to that point from earlier. Like in nature, there's succession where often these primary decomposer fungi get started breaking down stuff that's too complex for other microbes to go to work on. But it wouldn't get broken down into soil without the help of all those other microbes that come in, and then worms come in, and then birds drop seeds in, and plants start to grow. And it's that all of those organisms together having food and like having relationships that ultimately not only help address the contaminant, but make soil and make habitat and food and life that really is moving towards regeneration of a system. So, oh yeah, I threw this in last minute. This is just a link if you want to see the full report from the fungi farm study. And this is just their like findings. They did also note that they don't know how much they can trust their testing because, yeah, it's like we talked about, they really only, like a company had done the testing initially, so it's hard to compare. And then they only were able to test at the end, and they actually found a really uneven breakdown. So like, meaning they tested at a few spots over their treated site, and some areas actually still had really high levels of the hydrocarbons, probably because they were too wet, there wasn't enough air, the fungus wasn't able to do the breakdown in those spots. So one of their main proposals as well is to keep testing as you are working on a project. Test as much as you can before, during, after. That'll give you the best data. Okay, cool. So we're getting there. And then building off of, I just talked through a case study that I think demonstrates this point, that you really need to ensure that the fungus has what it needs to grow. And so that depends on what fungus you're working with, but you need to create good conditions for that fungus to live if you want it to put out those enzymes and do that work of remediating. And so every fungal genus, species, strain is different, but in general, fungi need moisture, oxygen or aeration, and some easier food sources. So that's why, the fungi farm study, they added straw and also cardboard in addition to the the diesel contaminated soil, to give the fungus a chance to like build up some energy and get healthy, to put out all those enzymes when they run out of that easier to digest substrate, to start to go to work on that diesel contaminated soil. And just that, like, as I think is demonstrated by the last two, which are some of the few like actual field studies that have been done, you see that there's a huge amounts of materials and resources needed. Like they had to like use big what are they? I'm totally blanking. They had to use like excavators and like big machines. They had to get a ton of straw and mycelium. So if you're working on a large site or with a large amount of the contaminated material, it can be really hard if you're using the approach of introducing a fungus that maybe isn't that suited or to that site. But either way, you need to consider, this is part of the making your plan, and also then installing and maintaining. It takes a lot of maintenance to maintain a remediation project and make sure it's successful. And so this diagram is also from the reading that I had sent, and it's just showing how these factors are all connected, like the properties of the contaminant, the properties of the soil or the site, and then the properties of the fungus or the organism you're working with. All there's an interplay that all affect like the success of degrading the contaminant. And so we had kind of hinted at this earlier. All that being said, you can also oh, Maya, do you remember what I think it was Larissa, somebody in the MycoRenewal course the other year had come up with an awesome way to describe training that is less like human hierarchical.
It was such a good word, and we wrote it down word and then forgot it.
Okay, well we'll open to ideas.
Facebook message Larissa right now.
Yeah, okay, cool. So yeah, because training makes it seem like we're doing something, but really like the point is that these fungi have all these amazing abilities and enzymes and this whole kind of toolkit, and we're kind of just supporting them to be more efficient at metabolizing a given pollutant or tolerating different conditions. And so I'm gonna just to make sure we round out the rest of the material, I'll just mention this for now. Really briefly overview it, and you can ask about it later when we get into questions if you're interested. But it usually involves just introducing a small amount, like an increasing amount of a contaminant to a healthy fungus, and giving it a chance to figure out like the enzymes it needs and figure out and get used to navigating that, and it gets better at it over time. So usually you just introduce an increasing amount of a pollutant to a healthy fungus. And then it's been observed that the strain that develops from that can be just more ready to go, more better at degrading a given pollutant.
The term I've been using there is facilitated adaptation.
Yes.
But it's not perfect either, and I'll let y'all know when I hear back from Larissa.
Thank you. So another thing people are talking about and working on at grassroots levels and corporate commercial company levels is actually just applying the enzymes of these fungi rather than living cultures. So there's obvious benefits because when you're applying just the enzymes, you don't have to maintain the site in such a way to support this living organism. And it's safer. So those concerns about introducing a non-native species aren't as much of a concern because you're just applying these enzymes. And it's worth mentioning that we already do this a lot. Like you can grow huge amounts of mycelium pretty easily. Mycelium is quite easy to grow, and then just harvest the enzymes. And this is done commercially, and a lot of like food products and all sorts of different products that we have are actually just the fungal enzymes that have been extracted from mycelium. So people are trying to figure this out on a DIY, more kind of grassroots scale. The problem is that enzymes degrade really quickly in the environment. And so what like Battelle I saw, like this big company is doing is they encapsulate the fungal enzymes so that they both are stabilized, so they can be like stored and transported and then get kind of turned on and activated when there's two triggers. So the fungal enzymes get activated by water and microbial, like other microbes interacting with them. So something to keep exploring. I've said this a bunch, but just to emphasize that, and it's come through in the examples, multi-kingdom, multi-phased approaches that mimic ecological succession are most effective. So I'll quickly mention like one study I've worked on, but usually going from primary decomposers to secondary ones, and then bringing in bacteria, insects, and plants is a good succession. And composting is something that's easy for all of us to do, no matter at different scales, and it's actually a really effective bioremediation treatment, especially when combined with mycoremediation. And then again, you need to treat the mushrooms that are any fruit bodies that form any like materials that are produced from your remediation effort as hazardous waste, and have a plan for how you'll contain or dispose of them. So I've worked on projects where we just had really like totally sealed containers where we continuously deposited any of our plants that had extracted metals, any of the mushrooms that may be fruited from the installation into these containers, making sure there's no runoff or anything. And then it's cool because they end up becoming this, like, going from this huge mass of contaminated stuff into a small amount of this like sludge that you can then, once you fill up your containers or whatever, bring to your hazardous waste disposal facility. So there's a lot of potential, and like I mentioned, like looking at the fungal enzymes as an option for organic contaminants, figuring that out on a grassroots scale would be really cool. Just trialing in the field all this cool stuff that we've heard might be possible, and then sharing that with people. Test as much as possible, partner with academic institutions, do good research, apply it in the field, and then share that with us. Share your mistakes, especially, because that's how we can all learn. Monitor projects over the long term. Make sure, as Kristen, I think, brought up, like, make it like monitor and keep testing and make sure that you're not like, woo, yay, it turned into soil, but actually there's, more recalcitrant toxic bits that have been left over. So especially like supporting, it's awesome that you're up in Alaska. I think, like, I'm from Canada. I think there's a lot of, I hope we all work on this everywhere. I think there's like sometimes when there, that's what I would like to really encourage. Like, try this out where you live, more northern climes, more rural places, remote places. We need to see how this stuff can be applied in ways that work in different places. It might be different everywhere. And so, and then share it with the rest of us. So because we're running out of time, I had a section on remediating toxic wastes. And, but I think maybe what I'll do is, do you think that I should try to just summarize one project in like 1 minute, Maya? Or, okay, cool. Okay, so this is all the same. My the model, this like mycodigestion of toxic waste that I've been working with, follows a lot of the same principles we've already talked about. The main thing is you have to collect it and contain it, and ensure the fungus has what it needs to grow. Follow those successional practices, and then be prepared to treat the mushrooms that may be produced as a toxic waste. So there's all these cool examples of this, from like large scale, degradation of disposable diapers in Mexico. There's a lot of people who teach and talk about this kind of stuff in different ways. One project I worked on was with a community bike shop where I used to live, and their bench tops and rags that are all soaked with these Teflon-based lubricants. And so I found out that this is actually like a really big issue, because a big percentage of the lubricants that we use on all sorts of machines and bikes wash off into the environment, and they stick around forever because a lot of them are Teflon based, and Teflon is one of those PFAS chemicals that's really bad for everything, and that's in all our blood and doesn't break down. And so really quickly, let me see, do I have another picture? Okay. Basically what I tried a whole bunch of different methods, but ultimately what worked out best was growing mycelium, and I used different types of oyster mushrooms as a primary decomposer to start the decomposition. I grew them on cardboard from the bike shop, which is awesome because it's free, it's another waste, and actually I found it way more effective than other types. So because you get these sheets, these planes of mycelium, then I would basically soak the lubricant soaked rags and bench tops and layer and stack this all in. I started with buckets and then scaled it up to these way bigger systems, like the barrels that you see here, like 50-gallon barrels. I made the same mistake at first, where there wasn't enough air exchange, and the mycelium didn't really thrive. So I did a lot. I kept working with the model, and ultimately got it to a point where I would do the primary decomposition with the oyster mushroom, and then put all of that material in a worm bin. And the worm bin functioned so the bacteria and the worms themselves worms love mycelium, and so they would physically and chemically continue breaking down all of that material. And it also gave me a sense that I was reducing the toxicity of these materials, because the worms could not survive if I just if I added just the Teflon lube rags in with the worms. So yeah, that's just one one proposal, and I think the model is something it'd be cool for folks to try out and work with, to deal with those more common but toxic wastes. And this is something that is very feasible at a community scale, because it's very easy to grow oyster mushrooms. They are effective at there's probably lots of others that would be effective too, but it is a contained way to deal with certain wastes. Use actually waste to grow the mycelium, and then work with the mycelium to start a process of breakdown. Finish it off with composting and other life. So yeah, and so I have a bunch of handouts on some of these different ideas for digesters on my blog if you're interested in trying that out. Thank you so much. So I think we still have a few minutes for questions.
I have a question. I was wondering about styrofoam. Like, if I have a styrofoam takeout container, could I bioremediate that? Have there been studies on that?
Yeah, well, actually, but the best, is it, what are they called? Wire worms or something? Does anyone know? What are those?
Oh, mealworms? I saw something about mealworms eating, like actually eating styrofoam.
Oh, yeah, like Oliver. So yeah, check out Oliver Kamenheller. He's really cool, and he works on some of that stuff. And I think it's a good, it kind of brings up a good point. Like sometimes fungi aren't going to be the right option at all. You know, sometimes like, you know, maybe gravel is going to be a better filter than King Stropharia, which, you know, or like maybe some weird worm is going to be better than a fungus. So just being really open to, and that's part of the research, choosing the right way forward. But you could, one thing you can try is literally just like go put pieces of styrofoam in different environments and then see what grows on it. And that could be a way to see if there are fungi or microbes that are interested in eating styrofoam.
Cool, thanks.
Yeah.
We have some questions here on the chat that I'm not able to answer. Okay. I think are more for you, Danielle. Does anybody want to ask their question that's on there?
I have a question. Chanel has a question. Hello?
Yeah, go for it.
Okay, sorry. I didn't know you could hear me. So just like we were talking about the having native and non-native, I just had, so in my backyard we have some fungi growing on the tree, but I haven't identified it. But just today we put the substrate of different oyster mushrooms, king mushrooms, and lion's mane. We put it in the bag. So I wanted to know, is that something bad, or is that something to like that we'll have to do further research in?
So sorry, it was King Stropharia oyster and lion's mane?
It was, I don't know, it was a blue oyster.
Cool. Do you live in North America, Chanel?
Yeah, I live in Brooklyn.
Oh, cool. Yeah, so my understanding is that a lot of the oyster mushrooms and lion's mane are from this continent, so they are like from here. I know that there are types of Stropharia that grow on different parts of this continent, but I'm not sure about King Stropharia. I know that it was used a lot in Europe, so I'm not sure if it got brought over from Europe. Does anyone know about King Stropharia, where it is from? So it's complex with fungi. I would be really curious about other people's thoughts on this, because fungal spores like end up like in the stratosphere and all over. They travel really far. But I think like looking at maybe more the potential impact, like it sounds really positive if you're growing food for yourself and, you know, making soil in your backyard. That sounds like a really positive thing. If, you know, if oyster mushrooms take over everything and mean that like there isn't space for other organisms, then that would be maybe something that would need to be balanced out. But I'm not, yeah, what do others think? Any thoughts on this?
I mean, my feeling is to work with native species where you can. I've gotten our local species of Stropharia called Stropharia ambigua growing on cardboard and wood chips pretty easily. And so that's something that I would rather work with if I was doing a mycofiltration project. But if I just want to be able to like buy a bunch of bulk spawn, you know, then I would just use, I think it really depends on like what resources you have available to you. But generally, you know, you don't hear a lot about invasive fungi really taking over or being that much of a problem. Largely because, you know, they just, like Danielle was saying, they just have their one part of the decomposition succession process that they take part in. So.
Okay, thank you.
I think a couple other people have posed the questions about glyphosates, but I'm interested to hear if you have answers about that. I think one of the question was like, how long do they generally take to break down? And then I forget some of the other questions, but it's just a commonly used one. And I wonder if there's some sort of a suggestion for a way to like treat with a glyphosate and then follow right behind it with some kind of inoculation that can help clean it up, or I don't know.
Right. Yeah, it's a good question. It's a hot topic right now because I believe like after the lawsuit with Monsanto, glyphosate is being banned. Is it being banned? Has it already been banned? I might not be up to date in the US. I would need to do a bit more research into that. We talk about it a lot in toxicology at school. It's really interesting because it's purported to break down really quickly in the environment. So a lot of the toxicity comes from like exposure through like to workers and to people in like on farms, and so that's like a really important thing. But I've been curious and I would need to do a bit more research. I haven't the fact that it breaks down quickly, I always wonder about that. I'm like, what do you mean? Does that mean that common native microbes in soils quickly eat it? Does it mean it is very leachable and like gets really easily like, you know, volatilizes or moves away in the water? I actually haven't seen, I don't have anything come to mind, but I'm really curious. I feel like this keeps coming up and I was like, oh, I should look into that. So I'd be happy to poke around and share some articles if I do find anything about like fungal, you know, treatments to degrade glyphosate. Has anyone seen anything about that? I know that a lot of fungi are used or like have been found in lab and bench scale studies to be able to degrade even like DDT, which is a very persistent insecticide. Glyphosate is commonly called Roundup and it's like a herbicide. But it would have a similar, I think a similar chemistry. So I would imagine that there are fungi that could do something with that. I just can't recall a study that I've seen about which ones. Yeah. Maya, you're muted.
Do two more questions, and then if we can move on, that would be so great. So there's a question here from Anne about hazardous waste facilities. Anne, would you like to ask that question out loud?
Sure. I'm revealing myself as a former hazmat inspector. So when you said that
cool.
you collect, when you collect the hazardous waste, did you have to educate the hazmat facilities that you take it to Is it small enough that you can take it to a household hazardous waste facility? I'm in California, so.
Thank you so much, Anne. So the projects that I was working on that I mentioned were in Canada and on Vancouver Island. And so I did, actually, it wasn't so straightforward to bring to a hazardous waste facility. But I did find I had some luck having some neat conversations, like depending on who was working that day or something, people would be like, oh, great question. Because I've worked on also phytoremediation projects, myco, bio. And so, they're like, what is she asking about today? I'm like, where do I dispose of these plants that are, they're like, just in your yard waste. I was like, no, they're toxic, So I did have to talk, explain and talk, and often the person at the facility needed to do some research and talk to some people and look into regulations and then get back to me. But that was all in Canada. I've just been in California for like a year and a half, so I'm learning. I'm less familiar with this system. Do you have any insight, Anne, for how we could best work with?
But it sounds like some education could be helpful.
Yeah,
yep.
Totally. Yeah, I do a lot of, so I end up making a lot of like 1-page like fact sheets. And those are really handy where you explain like what you're trying to do, why, and some of the info and places people can find more info. It's good to have that on hand when you're working on these projects.
It would be. I'm interested in doing a lot of lead remediation in inner cities around the country. As gentrification is pushing on neighborhoods, there's a lot of land that's been abandoned because it's lead contaminated. But if we could grow food locally, I was just stumped by what do you do with the hazardous waste when you grow mushrooms or spinach or whatever will take that lead out, and then you've got the lead out of the soil, but you still have a hazardous waste. So thank you. They're really helpful to know that somebody's actually tried to do this.
Yeah, and I will say, like, a lot of my pictures are really boring, but they're very exciting for me when, like, literally just pictures of like garbage cans where we've done phyto extraction of metals over a huge area over many years, and you end up with people growing food in healthful ways and just a small amount of like toxic sludge in the garbage can that's contained. Like so something as simple as that is totally feasible as well.
So Ellen had a question, and I do want to transition into the next part of this.
Okay.
And her question was around the molds and yeast that can be used for plastic degradation. Mm-hmm. And maybe, do you have like more resources that you could share around that?
Yes. Yeah, I think, yeah, for sure. And basically it's through hot composting. So those organisms are already present and they show up. And so effective hot composting is how those plastics were degraded. But you have to think about, I think we need to research if microplastics are being created through those processes, which can be toxic too. So I'll see if I can find other resources. Great question.
Awesome. Thank you so much, Danielle.
My pleasure.
Give Danielle some twinkly fingers if you like. A little silent applause. So I think, Danielle, if you turn off your screen sharing, we should be able to do the gallery view. All right, so now that you've done that, so if you go up in the top right corner, there's a little gallery view. And if you've been hiding with your screen off, it's very reasonable to do that on a call with all these people. But if you want, now is a good time to turn your video on so we can just kind of see each other and connect for a sec. Hi. So I wanted to take the next few minutes to just check in with folks and hear how this is going for you. And if you have any questions that have come up, if there's any holes in the knowledge that you don't feel like are going to be covered by we only have 2 more classes after this. And I'm curious to hear about how the homework load and the reading is going for you. And yeah, just if anything has come up that's like either, you know, something you really wanted to get out of this course that you are getting, or if there's anything that's surprising you, or anything like that. If folks could keep their shares, like if any of those questions, you don't have to answer all of those questions, but if any of those things that I just covered, it was like, oh, I really want to share this, and you can do so in less than a minute, that would be so great. Would anybody like to, you can raise your hand and I'll be able to see it. Would anybody like to share? All right, David, go ahead. You're muted. There should be a mute button in the bottom left. And I can try to unmute you. There you go.
Oh, great. Thanks for your patience. I was just going to say, I don't have the science background that many of you appear to have. So a lot of my effort has been building my glossary of terms, and it's been quite engaging. It's good homework practice, I suppose. But there's an awful lot of technical language and that I've had to get comfortable with. And so I just wanted to share that that is definitely a barrier for us who lack that grounding.
Got it. Yeah, that's really helpful to know. Is anybody else in the same boat? And you can also just email me later. So I'm wondering, is it, it's the fungal biology piece we covered on the first day, but I'm realizing that we could have done that a little bit more thoroughly, especially for folks who are new to this stuff. Do you think that would be helpful to provide more resources there and more, maybe have a dictionary, you know?
I think an agreed, I mean, as I encounter words over the readings, I'm constantly writing them down and then not sure where to go. I use Wikipedia a lot. You know, just whether there's an accepted place of reference or not.
Yeah. Well, the thing with mycology is everything is controversial and evolving and changing. So,
but it's good fun. I really appreciate the exercise of it all.
Awesome. Thank you so much. Would anybody else like to share how things are going, how the homework is, if you have any outstanding questions that haven't been addressed?
I'll go. I said, let's see. I was going to say that the homework on like making a design for a research plan for you know, how mushrooms can fix a waste in your community. I really liked that, and I think it's something that I really want to do. But in Alaska, everything's frozen right now, so I'll have to wait until the summer or the fall. Even though it's really sunny outside, I was just on the deck for a little bit. Today's a very warm day, but yeah, I like that kind of like concrete, like planning some designs, and there's a lot of ideas that it's kind of sparking, so I really appreciate that. And I also want to add that there is a mycoremediation project going on right now in Anchorage. I am not directly involved with it, but I was talking to another friend of mine who's a mushroom farmer, and she said, yeah, I have all this oyster spawn that's just waste, you know, every season. And so I need to put those two people in touch and be like, you should take that mushroom spawn and give it to this mycoremediation project. And that connection, I think, will help both of them a lot.
Beautiful. Thank you so much.
I want to second what Kristen said about the homework assignments. I'm kind of swamped for time right now considering the season, but it's something I plan on revisiting everything definitely and going through it that we've learned in the course. And just maybe a continued, and you're probably planning this already, but a continued contact list maybe could be helpful as I revisit things and, you know, take a little more time and probably have a lot more questions.
Yeah. Have folks been utilizing the contact sheet and reaching out and connecting, or cool. There's a study group that's formed.
I did have a question. As I plow through some of the documents that were being shared, I bump into paywalls.
Whoa.
And I didn't know if there was a person or a point or someone who I could share the link with and say, is there a way to get past this? I'm particularly interested in the salt marshes of Nova Scotia and some of the work being done at the Bay of Fundy, or the Bay of Fungi, as the folks at Acadia University call it, because it's such a unique environment. But quite a bit of that research into salt marsh and marine fungi ends up being behind paywalls.
David, I'm in school right now, so I have access. So feel free to email me,
Okay.
and I can send you some.
Yeah, I'm on Team Canada. Go, yay.
Yeah, are you? That's cool. Anyway, cool. That's awesome.
Okay.
Yeah, I think my emails, I'll drop it in the chat box, but I think Maya had sent it before.
Yeah.
Yeah.
Thank you.
Great.
My pleasure.
Awesome. I see Patricia's hand.
Yeah, I was wondering, because I've been looking at different online research, and it's so extensive. I'm wondering if you know about any places, because I'm mostly looking into degraded agricultural land and how to bring that back to life. If there's anywhere where I could like focus my like my looking, or if there's like sites that have more information on agricultural projects or things like that, like, or because I find a lot of little things here and there, but it's timely, the searching without having university access. Like, so do you know of any agricultural research sites or anything?
Like websites or physical sites?
Oh, like websites where they have research on agricultural, like mycoremediation projects or bioremediation projects.
Yeah. Well, part of the reason I'm studying that is because I think that linkage hasn't fully been made. I can certainly think about if there's certainly some articles about, like, mostly working with our arbuscular mycorrhizal fungi to help bring and then in combination with like other soil health practices. I'll think, Patricia, again, if you feel free to email me, and then I can respond with some links and suggestions for more agriculture focus, because that's totally my kind of background too. That's what I'm focusing on. So I'd be happy to share. I dropped my email in there.
Yep.
And the prompt
because it'd be nice to see how other projects are started, because agriculture is quite a bit of land, you know what I mean? So it's not like a small like, how do you do a whole field or acres? It seems like it has a whole different level of like how much mycelium there, you know what I mean? So it'd be interesting to see how other projects have worked it out.
Yeah.
And then what about costs? Like, is there any, we're just curious, like, well, how much are we going to spend to try to remediate this 5-acre plot,
It's purportedly low cost, but that's comparative to like other, like digging and dumping, like huge scale, like digging, like industrial remediation. It's considered low cost. For grassroots scale mycoremediators, that's why it's good to work with like non-toxic waste material, like organic matter that you can find as substrates that are free to grow huge amounts of mycelium on, and learn that skill yourself, or partner with a commercial mushroom farm to get spent kits, or just work on so yeah, and because there are still costs, but you can also fundraise. That's where community organizing and fundraising skills are helpful. I've done through partnerships and through applying for grants and through doing different types of fundraisers, been able to fundraise and work on projects for like between $500 and $10,000, which is a very big range, but it really depends on the scale of what you're working on and what your goals are and everything. But there's a way to do things at a lower cost, but usually through partnership and fundraising and grants things.
I have another question, if I can. I was trying to do some field chemical tests, and I found this one company that for like $90 you could do like 45 tests. But I had no idea, like, are they good? Are they bad? Like, do other people do this? Because somebody was saying, oh yeah, you don't need a mass spectrometer to test for these dioxins. That's what we're testing for in this other project. But there's chemical tests you can do, but it's like, well, these kits are so expensive and they give you like 20 tests. Like, do other people use those, or what, are you familiar with those kinds of tests?
Yeah, so I really think that like almost like bioremediation tool libraries are really cool ideas where you collectively purchase. So like you can get a HANBI is a field test for different chemicals I've used before, where it's $1,200 to buy it, and then after that each test is like $15. Supposedly EPA approved, pretty effective. Not all of them give you reliable results. It may not matter to you unless if you're working on a case where there may be a legal component, like if there's been a spill and that's part of it, you're gonna want to just pay for like testing that nobody can dispute, right? And so those field kits, they can really vary in their reliability. But public lab that I mentioned, they have or partner with universities who can just you can use, or like have somebody use the mass spectrometer for you and get good.
Yeah, it's tricky to convince universities to take on, unless you have a student that you can, it's tricky to get them to take on projects. So
right.
We're working with that now.
Well, I can be a partner. As I said, I can do metal stuff. I don't have equipment personally to do organics, unfortunately, but.
Okay, thank you.
Yeah.
All right. Thank you all so much. Folks are starting to drop. So I think we'll just end it. If you have any other feedback or thoughts or ideas about how we can support you better, please send me an email, amazonmyco@gmail.com. And thanks so much, Danielle. That was an amazing presentation.
Thank you. Nice to sort of meet you all. Okay.
Thank you, Danielle. That was an amazing talk. Loved it.
Yeah, thank you so much.
My pleasure. Yeah, reach out if you have other questions or want resources. Okay.
I may do that. That would be great if I could.
Yeah, I think we Danielle and I spoke about this, and I think in the next 2 weeks are the time to reach out to Danielle before she gets busier. So if questions come up, try to get her soon.
Cool. Oh yeah, okay, or just be patient, please. Just because I am in grad school and it's pretty full on.
Okay, I understand. Yeah.
Okay.
Cool.
Okay. Take care, everybody. Good luck with all your work.